Today, I have been asked to talk about necrotizing enterocolitis and its relationship with red blood cell transfusions. And as all of us have talked about on numerous occasions, the question is this the devil? Or is it okay if we go by the existing standards? And over the next 45 minutes or so, I'll try to, you know, present it to the best of my knowledge. And if there are any questions, then we can certainly, give some time to discuss it further. Um, I have nothing to disclose. There are no conflicts of interest. And these are the three things I will deal with. I'll try to spend the first fourth or so on the clinical evidence that we have, which has resulted into the clinical controversy that we have, that each one of us has when we try to treat an infant with a red blood cell transfusion. Over the next 30 minutes or so, I'll try to present the laboratory data that we have acquired over the past few years. And in the last 20% of the talk is exactly where do we stand? Have we been able to resolve this controversy to some extent? So the first question is, why did we start asking this question? What is the clinical evidence? And in the next few slides, I'll try to summarize as to what we know. And to start with, I think the best way is to start by looking at the meta-analysis of pooled data. Mohammed and Shah from Canada first reviewed 11 observational studies. So these are all observational. And the data showed increased odds of developing necrotizing enterocolitis within 48 hours after receiving a blood transfusion. In meta-analysis of 5 studies that they found to be eligible for inclusion in this statistical analysis, they noted increased odds, almost fourfold, which were statistically significant. In another study, Tao and his co-workers reviewed 7 case-control studies, and there were 480 cases of transfusion and a fairly large number of controls. The odds ratio was still increased in the next 48 hours, and they found it to be almost fourfold. Most studies with negative effects of transfusion were retrospective, observational, and were limited by a small number of subjects that were looked at. Several studies showed conflicting results, adding to the controversy. And more recently, there was another meta-analysis where Garg, who is at University of Mississippi, he analyzed 17 studies, so a much larger number of these studies, and they did not find any increase in the risk of necrotizing. Another meta-analysis by Rai and his colleagues with 10 studies, they actually found a 45% reduction in the odds of developing necrotizing enterocolitis after a recent transfusion. So fairly controversial. In other studies, Sharma and his co-workers, they compared prospectively, they compared 42 cases who developed necrotizing enterocolitis versus matched controls at 3 centers. It was a small N, but they saw no association of transfusions with necrotizing enterocolitis in the following 48 hours or in the 7 days after this transfusion. In another study, El Abiyad and his co-workers, they reviewed 174 cases with NEC. 116 had received transfusions. The transfusions were not associated with necrotizing enterocolitis in babies who were born with a birth weight less than 1,000 grams, but they remained a risk factor in those born between 1,000 and 1,500 grams. In another study, Ravi Patel from Emory University in Atlanta, he evaluated nearly 600 very low birth weight infants it was a secondary but prospective, multi-center, but observational cohort. They noted necrotizing enterocolitis in 44 infants, which was about 7%. It's representative. And at 8 weeks of chronological age, the incidence of necrotizing enterocolitis was higher in infants who had received a transfusion versus those who had not. The incidence of NAC was 9.9% versus 4.6%. So it was a difference, it was a significant difference. They associated necrotizing enterocolitis with severe anemia. The hazard ratio was almost 6. It was highly significant, but they did not find an association with RBC transfusions. So if we look at the clinical part, it looks fairly confusing. Then there are other clinical studies. When we look beyond red cell transfusions, you can look at recombinant erythropoietin or one of its longer half-life derivative, darbepoetin, which may reduce the need for transfusions, and they found a trend towards protection against necrotizing enterocolitis. In another study, Olson and Aher, they studied nearly 3,600 infants. These were all low birth weight, and one group received early, like before 8 days, erythropoietin and darbepoetin, and they compared this group of infants with those who did not receive EPO or darbepoetin, but had received placebo. Early administration of erythropoietin reduced the need for red cell transfusions and donor exposures. And in a sub-analysis of 15 studies reporting 2639 infants, a fairly good number, the administration of erythropoietin or darbepoetin reduced the NAC incidence of NAC. The risk ratio was 0.69, and the number to treat to benefit was 33, so not too bad. So we developed an animal model in our lab, and we took 4 groups of animals. The first group was animals which were just observed. This was a group of naive mathematical controls, and these animals were just observed. And we observed these animals for 13 days. The second group of animals who were otherwise normal, but had received a blood transfuser exposure. And this was given on postnatal day 11. The third group was a group of mouse pups which had been exposed to venous phlebotomy on postnatal days 2, 4, 6, 8, and 10. And by doing that, we rendered these mouse pups to develop severe anemia. That is, their blood hematocrit was between 20 and 24%. So these were severely anemic mouse pups. And in the fourth group, these severely anemic received a blood transfusion which was given after the 4th day in an attempt to correct the severity of anemia and to simulate what we see in the clinical situation. And what we found was that in mouse pups which were in the naive control group, in the transfusion control group, or in the anemic control group, the intestine remained normal. But in the 4th group of anemic mice, which received a transfusion, they developed intestinal injury in the distal ileum, in the cecal area, and in parts of the large intestine. We looked at these samples under the microscope, and what you see is that the naive control were normal. This is the small intestine. This is the large intestine. Mouse pups which just received a transfusion were also normal. So no injury in small or large intestine. Then in mouse pups which just had anemia, had some degree of inflammation, and I'll show that in subsequent slides, in both the small intestine or the large intestine, but there was no mucosal injury. There was inflammation, but no damage. And in the fourth group, these were anemic mice which had been treated with red cell transfusions. Those mice developed histopathological evidence of injury, both in small intestine and in the large intestine. And we often describe intestinal injury in four grades. In grade 1, it is only mucosal injury. In grade 2, you have submucosal injury. In grade 3, you have involvement of the muscularis layer. And in the grade 4, there is a transmural injury. So these are grades 1, 2, 3, and 4. And what you see is that there was intestinal injury only in mouse pups that were anemic and had subsequently received a RBC transfusion. This is measurement of fatty acid binding protein number 2. It is a protein that is released from the intestine during intestinal injury. This can be measured in the bloodstream and can be measured as nanograms per milliliter. And you see that this FABP2 is increased only in the anemic transfused mice, meaning this gives further evidence to what we see in histopathology in this slide. And again, to emphasize that the histopathological evidence of injury was only in this fourth group. When we looked further, I said we noticed some inflammation in anemic mice. So we started looking at the number of leukocytes just under the microscope. And what we found was that there was no inflammation, obviously, in untreated mice. Surprisingly, there was also no inflammation in normal mice which had just received a red blood cell transfusion. But there was inflammation in anemic mice. There was increased leukocyte infiltration, but did not have any obvious mucosal damage. But the fourth group, which had mucosal damage, also had inflammation. So inflammation was seen in anemic, but with no mucosal damage, and anemic transfused mice, which also had mucosal damage. So these two had inflammation. This came as a surprise. When we look at histopathological sections from these two groups, what came as a surprise was that the inflammatory cells had the appearance of macrophages. When we look at macrophages in histopathological slides, they're often nuclei with a single indentation, like a bilobed nucleus, and with a large amount of cytoplasm, which appears pinkish, eosinophilic. And you find a large number of monocyte macrophage-like cells in these tissue sections. So we started looking at these monocytes macrophages. And this may appear complex at the first look, but I'll explain everything. So we first looked at mouse pups, which were in the naive controls group. That means these mice or mouse pups were normal. We hadn't done anything to these pups. And when we look at these cells by flow cytometry, then these cells, each dot represents a cell, then these dots actually reflect myeloid cells or white blood cells. And these have this antigen called CD11b. We then took these cells and looked for a macrophage marker. And these three groups, these are called the resident macrophages. This group represents the newly recruited macrophages, which come from blood monocytes. And these are non-macrophage cells. This is towards the negative side. So this does not express any marker for macrophages. This is naive control, mice which had no intervention. The second group is normal mice, which had just been treated with a blood transfusion. And when you looked at this, and we just took the same subgroup of cells, you found only the same subgroup. You found that these are F480 positive macrophages, but nothing else. So transfusions by themselves was not poison. The third group was just anemic mice. These had not been transfused. But you see that the number of white cells had increased. And now when you analyze this population on the right side was still there. But now you had an additional population where these white cells came in. And these are typically called F4AD, which is a macrophage marker, but F4AD mid. And in immunological studies, these cells are typically considered to be monocytes. The fourth group is, and you see the increase in number, these are now 36%. And when you transfuse these anemic mice, then you still found this upper population to be high, and you still saw a further increase in the total number of macrophages from 36 to 49, and you still had this middle population. These newly recruited, so these are macrophages, F4AD is a macrophage marker. These are macrophages were the cells which were originally in anemic cells. And these red cells they comprised of the original cells and also the cells which came in. These are derived from newly recruited monocytes. And you see that these guys in the red bars, they expressed all kinds of inflammatory markers. So the characteristics of these cells had changed. These cells now became activated and they became inflammatory. This is the fold change in gene expression. So if you see, the higher bars are the ones which are inflammatory. Then we wanted to investigate these further. On the first look, these slides may look complex, but they're not. So these mice are naive control, and the white blood cells are only towards the right. These are the cells which are native cells. They were there in the intestine always, in normal mice. Then there is a drug called clodronate. If we administer clodronate, then we largely remove these cells from the mice. These specifically deplete this cell population. In the anemic transfused mice, you see that the number of cells in this column and in this section have appeared. This has increased. These are the newly recruited cells. And then when we give clodronate, which is the chemical that depletes a mouse of this middle population, these macro-inflammatory macrophages, you see that we achieved our ambition, that these cells were decreased, but this new population disappeared. We measured this, and you see this red line is the transfusion control. The blue line below that is the anemic control. So nothing happened there. But in clodronate control, there were no macrophages. And when we give clodronate and anemic transfused mice, you were able to reduce mortality. Here I have mentioned as physical distress, not mortality, because by rules, in mice that develop physical distress, we take that as an early indicator of intestinal injury and we sacrifice these animals. And you see that in the mice which had been treated with clodronate, those who did not need to be sacrificed, only the anemic transfused mice, they needed to be sacrificed or they died spontaneously. And this is showing the same information by a different way, that when you treated the anemic transfused mice with clodronate, the grade of injury was significantly lower. So macrophages were the cells which caused intestinal injury. We wanted to confirm that further. And this slide might appear complicated, but again, I'll walk you through it. It is not that complicated. One of the markers of macrophages is the colony stimulating factor receptor 1 gene. This is expressed exclusively on monocyte precursors or monocyte colony stimulating factor receptor. In the exon 21, exons are the sections of a gene which eventually help in production of a protein. We modified this exon and we inserted a stop codon. And this is an internal ribosomal entry site. Don't worry about it. This is a diphtheria toxin receptor, and this is a red marker which we inserted. So these mice are genetically modified. Just let's go with that. When we cross this mouse with another mouse which expresses this particular genetic sequence, then you selectively cut out this stop codon. And these three genes start becoming expressed. And this is what I'm saying here. When you do that, then you modify your mouse. So these are the mice which were, which were genetically modified, but these genes become activated only when you administer diphtheria toxin. And in these mice, when they're anemic and have been transfused, you see the two populations. When you administer diphtheria toxin, you modify these mice genetically and you remove the middle population of inflammatory macrophages. When you do that, you reduce the mortality. So this complex looking slide, all it shows is that when you remove this middle population, then you reduce mortality. So the mortality is occurring from newly recruited monocyte-derived macrophages. You see this by another means. We have immunostained these intestinal sections. The green dots are the native macrophages. The red cells are they're enlarged, elongated, meaning thereby these were activated. These are monocyte-derived macrophages. And these red cells are seen only in anemic or anemic transfused. So these cells were recruited. You see that these cells were recruited in anemic cells, but transfusions were required to activate these cells so that the tissue got damaged. You see these cells that were drawn out of these animals. In anemic, you saw these monocyte-derived macrophages. But when you transfuse these, you see all these pseudopodia, which means that these cells actually became active. This is another way to show activation, that these cells which look green, they also started expressing this red cell, which is a marker of activation. In the anemic cells, the cells were there. You see these green dots. These were all macrophages, but they did not express red color. So that means these cells were not activated. But in anemic transfused, it was the transfusion which activated these cells, and now they start showing both red and green, which is seen as yellow. We showed that by different methods. We isolated these cells, and NF-kappa B is a marker of activation. And by various methods, we showed, recombining everything, that it is the red cells which activate these cells, these monocyte-derived macrophages. We showed that further, that it was the heme component in the transfused cells, transfused red cells, which were causing this damage. We again showed how, and it was that the hemoglobin that was derived from these transfused red blood cells, they activate toll-like receptor 4, the same receptor that is activated by bacterial endotoxin. So all of us know that the bacterial flora, that is actually what activates the inflammatory pathways. But what we found in this study is that it is the hemoglobin that is released from stored red cells which activate the same receptor that is activated by bacterial endotoxin and causes intestinal injury. Another way to say that, this is further evidence that the red cell transfusions activate toll-like receptor 4 the receptor for bacterial endotoxin, and that causes injury. This evidence is from mice that have been genetically altered so that they don't express the toll-like receptor 4. And when we did that, then there was no injury. Another way to show that, the green shadows are show activation of NF-kappa B, which is an inflammatory marker. And when we block NF-kappa B by giving nanoparticles, then the injury goes away. Then we protect these mice. Just multiple ways to show that the pathway we were looking at is real. And again, this is something that is clinically relevant to you and me, that this transfusion-induced injury is much more severe in pups with severe anemia, but was lesser in those with moderate anemia. And I'll come back to this. Then it is the duration that you spend in severe anemia. So if you allowed these mouse pups to remain anemic for 24 hours, injury is more. If you transfuse sooner, injury is less. You could also ask that, hey, if I wash my red cells, would I reduce the level of injury? And indeed, if you removed the white cells and you washed your red cells, the red cells you transfused, the injury was decreased. Does the duration affect? The conventional intelligence is it should, but we did not find significant evidence. So the consideration that you reduce the number of donors might still be valid. So you take red blood cell from one donor and use the same donor to transfuse again and again. As long as your red cells are not damaged, you may be okay, but it needs further study. Does the number of transfusions affect? And yes, indeed. If you give more than one transfusion, your risk is higher. And this is being shown in a different way. This is the number of transfusions, and this is the grade of injury. So the severity of injury also increased as the number of transfusions increased. Now I go to the third part, that I have clinical evidence which was uncertain. Then we looked at the animal model, which looked convincing. So where do we go from there as clinicians? One is transfusions by themselves were not injurious. So maybe the use of the word transfusion-associated NEC needs to be modified. And maybe it needs to be called transfusion and anemia-associated NEC to emphasize the importance of severe anemia. The second thing is that our current evidence favors minimizing the exposure of premature evidence, potentially at high risk of NEC, to prolonged and severe anemia. At present, there are 4 sets of national guidelines that are available, and the risk of transfusion seems to increase with chronological age. These guidelines are from Britain, they're from Australia, Canada, and one another country in Europe. But it is the chronological age which is an important consideration. We do need to identify safe hematocrit or hemoglobin thresholds for RBC transfusions. How anemic can we let them go? We also need to evaluate, once again, the potential benefits of erythropoietin or darbepoietin, at least in specific subsets, like babies with congenital cardiac defects, if not in every at-risk infant. Only a minority of severely anemic premature infants develop NEC, and so there is a need to identify genetic risk factors or clinical risk factors, because not everyone who is severely anemic develops necrotizing enterocolitis after receiving a red cell transfusion. Considering the typically delayed onset of anemia of prematurity after birth, studies are needed to identify postnatal periods of increased vulnerability for more intensive monitoring. You cannot put multiple monitors on every premature baby, because then you're reducing the opportunities to improve maternal-infant bonding. If the baby has 15 monitors attached to him or her, how do you do that? But if you define a chronological period in which the baby is at higher risk, then it may be possible. It may be a smaller cause. Then we need to identify infants with ongoing subtle subclinical hemolysis. These infants may have anemia, but they also might have activation of monocytes and macrophages and the need for repeated transfusions. Everything we have found to be associated with transfusion associated with anemia and NEC. Again, to show it pictorially, it is the free hemoglobin, which might activate toll-like receptors in the intestine. It is the free hemoglobin that is important. It is not the red cells that may be important. And this free hemoglobin might activate blood monocytes or intestinal macrophages, and these two sets of blood cells activate and damage the intestine. Now, what about vascular autoregulation in the intestine? So splanchnic vascular autoregulation needs further evaluation. And suddenly we're in a position where handheld or pocket-held ultrasound machines might enhance the feasibility of this. Association of transfusion and anemia-associated NEC with stimuli that increased intestinal oxygen consumption, like feeding, might need evaluation yet again. Transfusion thresholds of hematocrit or hemoglobin are still unclear. Animal models suggest 24%. But remember, mice don't have fetal and adult hemoglobin. So some human studies are still needed. And some human studies that I did with collaborators in St. Louis, they actually suggest that the intestine may be at risk of 24%, but the brain might actually be at risk at 28%. So the story is not done. We need more. We need to view red cell transfusion thresholds as a range, not exact levels of hematocrit or hemoglobin, because the corrected gestational age, altitudes of residence, like my collaborators in Utah, which is at a higher altitude, they may be at different comorbidities and the functional status of the microcirculation, because you might have a small for gestational age baby who might have altered microcirculation. The rest may be completely different. Then murine studies do not show harm in transfusions with red cells stored for short periods. These findings are at variance from what we know currently, that RBC storage is a bad thing. We need to look at it yet again. Murine studies show no harm in transfusions with short periods. And these, again, I'm repeating this to emphasize this, that these findings are at variance with existing evidence from human studies. Need to look at it again. The concept of anemia in premature infants needs evaluation. Fetal and adult hemoglobins have different oxygen carrying capacities, and so low hematocrit levels in an infant who has previously received red cell transfusions from adult donors may have completely different physiological implications than another infant who is not. The advantages and disadvantages of washing red cells needs another reevaluation. Washing may reduce the half-life of transfused red cells, but it may remove free hemoglobin and the red cell contents to reduce the risk of NEC. And that's where we need to look at mouse and human studies very, very carefully, because mice are not men. Severe anemia may also affect the brain. The intestine is not everything, and follow-up studies may need to carefully examine the outcomes of these infants in comparison to those with NEC related to other predisposing factors. We also need further study to confirm that transfusion-associated NEC in human infants is more likely to involve the ileocecal region. And when we consider that transfusion and anemia-associated NEC could be delayed, the colonic region needs to be very carefully assessed. The relationship between enteral feedings and transfusion and anemia-associated NEC needs to be carefully evaluated. Even though observational in design, there is a beautiful study going on in the United Kingdom. It's called the WHEAT study, withholding enteral feeds around packed red cell transfusions. And these studies like this may yield very important insights. And nutritional interventions, pasteurized human donor milk, enteral substitution of lactoferrin or L-arginine, and preventive application of multiple strain antibiotics. I think these need to be examined very carefully with sufficient monitoring, such as with ultrasound. This is where I'll stop. I wanted to thank some of my collaborators and colleagues. They're here, and this is here where I'll stop. Thank you so much for giving me this opportunity.